And thank you all for being here. I'm really excited to finally get to talk to you all about some lions in, again, my dissertation entitled A Century of Conservation Genetics of the Lion Panthera leo. Lights. Hey, can you see it better now? Okay. We all know what a lion is. And not just because of The Lion King and the remake coming out next month. Shameless plug. No, for centuries, the lion has been used as a symbol to represent bravery, courage, pride, and strength. And growing up, when we were learning about animals, we all learned about the king of the jungle. Now, this is a map of the historical range of the lion. The lion is a very versatile species. It can exist in almost any habitat except the Sahara Desert, and interestingly enough, the deep jungle. And I've already lost track of my thoughts. All right, so all right, so over the past century, the lion has been pushed out of its historic range due to anthropogenic changes to the African landscape. So there have been 23 subspecies of lion recognized throughout history, but currently there are only 2 subspecies of lion recognized, and the definition of those subspecies is determined by which organization you're referring to. So CITES and IUCN both recognize an African and an Asiatic subspecies, where the Asiatic subspecies has a higher listing and more protections than the African subspecies. But U.S. Fish and Wildlife in 2016 passed legislature within the Endangered Species Act, which moved West and Central African lions with the endangered Asiatic lion. This decision was based on a number of mitochondrial studies, which showed that the Western and Central African lions are more genetically related. As you can see here, here, and down here, genetically related to the Asiatic lion. Now, this was only based off of mitochondrial results, so since there weren't nuclear results that went along with it, IUCN and CITES didn't believe this was enough scientific evidence to change their designation. However, the U.S. Fish and Wildlife decision is more in tune with the conservation needs of the lion, as in recent years, the Western African population has taken the hardest hit. So the major sources of lion mortality are all anthropogenic in nature. They're human encroachment into lion habitat, habitat destruction and land conversion, reduction of the prey base, and retaliatory or preemptive killing, all associated with increased urbanization and rural expansion as the human population increases. So in the late 1880s marked the beginning of the Scramble for Africa, which was the beginning of an exponential increase in the human population in Africa. And as the human population continues to increase, the lion population decreases. And the human population in Africa is just projected to keep increasing, suspected to quadruple by the year 2050. And a lot of conservation decisions are based on the extent of population decline. But there's been a lot of debate about population estimates with the lion, whether they're coming from scientific estimates or from the media. The current scientific estimate of the lion ranges from 16,000 to around 40,000 individuals. And this is because it's a collection of local studies that have made estimates based off of using different techniques at different times. And the extent of decline is because that number is compared to a historical number from a magazine article from the 1970s, which stated that there were more than 400,000 lions in Africa prior to 1950. Now, this number is based purely on speculation, and there's been no attempt to validate its accuracy. So we don't actually know the true extent of population decline. What we do know is that their habitat has become greatly reduced. So rather than focusing on exactly how many animals are left in the wild, we should be focusing on if lions are genetically healthy and if genetic diversity is declining. And are lions being managed in a way that's beneficial for the population? So keeping those questions in mind, the objectives of my dissertation were to create a baseline for genetic health so we can have something to base that decline off of, and to document changes in genetic diversity. So there are three main parts to my dissertation, which I'm going to go through with you one by one. The first is developing new protocols that were more appropriate for the species as well as the sample type. So all previous genetic research of the lion has been based on genomics of the domestic cat. Cross-species amplification is a common practice in conservation because it's convenient to use protocols and resources that are already developed for a highly studied related species. And for the lion, that's the domestic cat. But the more diverged two species are, the more potential for issues downstream. And the domestic cat lineage and the Panthera lineage are approximately 10.8 million years diverged. It's a lot of time to introduce change. In the lion, there have been 12 studies that have used microsatellites, but there's been no consensus for microsatellite use. Out of 107 microsatellites, there's only a single locus that has shared between all 12 studies. And in 2014, Miller et al. Evaluated 31 microsatellites to determine which ones would be better used for population genetic studies or for forensics, but they didn't go and take that step further to assess for divergence or attempt to improve upon the loci, even though some of them did not perform as expected. So I developed a panel of LEO STRs by redesigning primers from previously used microsatellites. Primers were redesigned to be specific to the lion and closer to the target region, which is important when using potentially degraded DNA, which is often the case with wildlife studies and using museum specimens. And then by using previously used microsatellites, this allows for the continued use of previously published data, assuming proper calibration of allele calls across studies. So I chose 17 microsatellites. Based on how many studies they were used in, they had to have high heterozygosity and that they were highly polymorphic. I then cloned and sequenced 3 lions for all loci, and they used those sequences to compare to the domestic cat sequence and for primer redesign. So in comparing the domestic cat to the lion, there was actually only one locus that was identical between both species, other than repeat number. And what we saw were differences actually in repeat motif. And in this graph down here on the bottom, this shows point mutations between the two species. So as you can see, there's a lot of differences. So that warrants doing this primer redesign. So the final panel was 14 LEO STRs. 3 of them were removed, of those microsatellites were removed because one could not be cloned. One was not a microsatellite when it was sequenced, and the third, we couldn't find a primer pair that fell within our criteria using the sequence. Those 14 LEO STRs were multiplexed into 4 mixes, and those mixes were tested on 30 animals to test for efficiency, and they resulted in 100% amplification success, and they had high heterozygosity and were all highly polymorphic. Now we have those protocols. I wanted to focus on a local population of lions in Zambia. Some studies have suggested that the evolutionary cradle of the lion is in Southeast Africa, or this yellow shaded region. Yet there hadn't been a local study that had focused on this area. And range-wide studies either had low sampling of less than 10 individuals from a single national park, or missed this potentially core population completely. Now, Zambia is interesting geographically in that it is the transition from the Great Rift Valley into the southern veldt, or the southern grasslands. This is a soils map of Zambia, and if you overlay the areas that have the most lions, you can see that the east is very different from the west. In the east, you have this purple and green, which is the end of the Rift Valley. And over here, you have the brown and orange, which shows the beginning of that southern belt. So not only are they geographically different, but they also have an anthropogenic barrier in the form of a large city, the largest city in Zambia, Lusaka, which is surrounded by an anthropogenic patchwork of towns and farms, which have long been considered uninhabitable by lions. So in collaboration with the Zambia Lion Project, who collected collected samples from 409 lions from 2008 to 2013, we wanted to take a closer look at the population of lions in Zambia to assess genetic diversity, population structure, and the potential for movement of lions across the country. And we did this by looking at nuclear DNA and mitochondrial DNA, by genotyping those 14 Leo STRs, and sequencing an approximately 1,800 base pair region of the 12S to 16S ribosomal genes. I was able to genotype 390 of those 409 lions, and structure analysis gave us results that were fairly expected. 2 subpopulations, one in the west and one in the east. What was a little less expected was when I calculated the number of migrants, and that came out to be 5.6 migrants per generation, indicating there is some movement between the 2 subpopulations. So I did a test for isolation by distance through a mantle test, which takes into consideration genetic distance as well as physical distance, or geographic distance. And that resulted in low but significant isolation by distance, which you can actually see in the factorial correspondence analysis, where it shows the 2 main subpopulations with the pink and the blue, but then there's these intermediary individuals. And when we look at the map, those intermediary individuals are from the corridor and lower Zambezi. So the likely movement between the 2 main subpopulations is through this region and through this anthropogenic patchwork. So we see further evidence of that eastern and western subpopulation when looking at population statistics in evidence of a Walland effect. A Walland effect is when you assess the population as a whole, there appears to be a reduction in heterozygotes. But when you separate them out into their 2 subpopulations, those subpopulations are each in Hardy-Weinberg equilibrium. They also have a high number of private alleles within each subpopulation, with 31 in the east and 14 in the west. This structure is also seen when looking at the mitochondrial DNA on this, and this is the haplotype network. And you can see that the western haplotypes cluster together with the eastern haplotypes. You see that there are a few individuals that what we called were crossovers, and each one of these individuals were male, and so likely a recent migrant. We also found evidence for the evolutionary cradle. And Tunis et al. in 2008 described 12 haplotypes across the range, but they did not, when they sequenced 357 animals, but they didn't sample any individuals from Southeast Africa. So when we added 391 samples, all from Zambia and Southeast Africa, that added 6 novel haplotypes, and we also observed 3 of those previously described haplotypes. So Zambia actually shows some of the highest diversity values for both mitochondrial as well as nuclear DNA. And that's more than any other local population that's been studied so far. Now, an AMOVA can tell us how much gene flow there is between 2 subpopulations by calculating an FST value. For mitochondrial DNA, the FST is relatively high, meaning there's little to no matrilineal gene flow between those 2 subpopulations. Now, while for the nuclear DNA, those STRs have a relatively low FST value, indicating greater nuclear gene flow. This is likely because of these 5.6 migrants per generation are all male. Now, lions exist in prides, where there's multiple females and 1 to a few males, where the females stay in or near their natal pride and males disperse. So it's this mating system which causes this discrepancy between the mitochondrial, the only maternally inherited gene flow, and the nuclear biparentally inherited genes. So now that we've filled this gap of knowledge in a potentially core population, modern population, good job, Linda. Now I'm going to move on to the comparative study, so we can look at how has the human population influenced lion genetics. Well, the best way to do this is by doing a direct comparison using a historical population to a modern population. So during that time of the scramble for Africa, that also was a time of an increased desire to have ample collections at natural history museums. So turn of the century, explorers, hunters, and naturalists scoured the planet looking for specimens to include in these natural history museums. And in doing so, collected hundreds of lions, which are now being preserved in museums across the world. So our historical data set consists of 162 lions collected from 11 museums across the world. These 162 individuals are from 1895 to 1930. So DNA was extracted from dried tissue, hide, bones, and teeth. They were genotyped on those 14 STRs, as well as whole genome sequenced to get their mitogenomes. Now, the modern data set is building on existing knowledge by using previously published data from 3 studies. Driscoll et al. 2002, Bertola et al. 2015, and the Zambian study. Now, these 3 studies have overlapping sampling, which was important when it came to calibrating the allele calls across the studies. So between these 3 studies, we have a modern population of 135 individuals, range wide from all of these different locations listed here. So when using samples from 2 different time periods, you have an issue of labeling, because Africa has changed greatly over the past century. There are some countries that have changed their name multiple times, such as Leopold's Congo, which became the Belgian Congo, which is now currently the Democratic Republic of Congo. Then there's also areas that have completely changed their borders, such as British East Africa, which is now Kenya and Uganda. So to make labeling consistent, every single sample was categorized into conventionally recognized regions that made West and Central Africa are our Western population, East Africa is the Eastern population, South and Southeast Africa are our Southern population, and then North Africa all the way over to India are the Northern population. And then as you can see from the sampling map over here, where the green dots are the modern population and the pink dots are the historical population, we have fairly equal sampling across the entire range between the 2 data sets. So for nuclear DNA analysis, there were 9 overlapping loci. This means they were present in both data sets, and they had greater than 75% coverage across all the samples. So once individual samples that had too much missing data were eliminated, that left our modern data set with 135 lions and our historical data set to 137. We also whole genome sequenced 153 lions. Those reads were mapped to a lion reference sequence that was provided to us from Stanford, to which we added 2 additional scaffolds, a mitogenome sequence and a representative new mite sequence. Then after some conservative filtering, we had 102 historical lions and 3 modern lions left over, mitogenomes, to which we added 16 modern mitogenomes from NCBI. And after multi-sequence alignment, went too fast. We were left with 280 polymorphic sites, which is what we used for further analysis. First, I did structure analysis of the nuclear DNA, and knowing that regional studies have found structure, or there have been structure found in these regional studies, such as with the Zambian study, I implemented a hierarchical strategy. This means that you run structure for each population determined by delta K until no more structure is found, and this should find any fine structure within the population that might be missed if you just run it all as a whole. So for the modern population, this resulted in 4 tiers of structure, which can be viewed at delta K, or which can be viewed at K equals 2, K equals 4, K equals 6, and K equals 11. So the first tier separates our Asiatic lion from the African lion. The second tier, those colors might look familiar to you. This is our conventionally recognized regions. So you have north, west, east, and south. The fourth tier breaks down that southern region into a southwest, a true south, and a southeast. And then the fourth tier brings you down almost entirely to the sampling population, to a local level. In fact, the only area was Tanzania where you couldn't identify each individual sampling location. Now, this is in stark contrast to what we found for the historical population, which I think is best visualized through the principal coordinate analysis. Now, on this side, on your left, you have the historical population, where these dots look seemingly random. And this is likely because it's a panmictic population. Whereas on your right, you have the modern population, and you can see that subcontinental structure. So now referring back to that mating system, remember the males disperse. So without barriers, without anything stopping them, males can travel thousands of miles in a generation looking for a new pride and breeding. So it is very possible that a historical population that doesn't have fragmented habitat could be panmictic population. Now, the mitogenome structure looks a little bit different. Now, it didn't matter whether you were looking at the haplotype network, the phylogenetic tree, or principal components analysis. Both identify 4 different clusters, or 4 different clades. We have a, pardon me, oh my goodness. There's a southern, an eastern, a mixed, which has haplotypes from individuals that are both in South Africa as well as East Africa, and then a western clade that has the Asiatic lion embedded within it. Now, if we compare that to some previously published haplotype networks on regions of the mitochondria, 12S to 16S, and 2 different site B sequences, we see the exact same pattern of south, east, mixed, and west with the Asiatic lion embedded, indicating that it's likely that the mitochondria has been maintained over time. So if we take a closer look at the maximum likelihood tree, bootstrap support greater than 70 is shown, and the nodes that separate out these 4 clades have some of the highest support. I want to bring to your attention these 4 blue dots that look like these 4 blue haplotypes that look like they might have gone rogue. But if we go back to that labeling map, each one of these individuals are from Gabon, Congo, and the southern Belgian Congo, which is now the Democratic Republic of Congo. And that is all of this lower blue region here. So it does make sense that they would cluster with South and East Africa, as opposed to West Africa, where they would have to cross that jungle of a barrier. So we have our 4 data sets, the historical, modern, and nuclear DNA, and mitochondrial DNA. What does it say when we put them all together? Now, for the nuclear DNA, heterozygosity is high for both data sets at around 0.8. So if you were going to look at the modern population by itself, you'd think that's really high heterozygosity. That's a healthy population. But when you compare the two, there's actually been a significant decrease in nuclear diversity from the historical to the modern population. Now, the mitochondrial DNA, those mitogenomes, have identical haplotype diversity. They're exactly the same, showing that there's been a maintenance of mitochondrial diversity, as we also saw in the phylogenetic tree and the haplotype network. So what do we have? It's male-mediated gene flow interrupted by habitat fragmentation, resulting in increased fine nuclear structure and decrease in genetic diversity as subpopulations become more differentiated as these fragmented areas become more isolated. And while mitochondrial structure and diversity is maintained over time, primarily due to that mating system where the females don't go anywhere. So a lot can change in 100 years. The scramble for Africa increased European rule from 10% to 90%, marking an exponential increase in the human population, urban development, and rural expansion. This inevitably led to a reduction in the lion's habitat, causing fragmented and growing populations that are becoming increasingly isolated, which results in subpopulations that are becoming more distinct. So now we have, this is kind of a big deal. We now have evidence of population decline and changes in population structure of the lion in a species that otherwise looks like it's doing pretty well. So what does this mean for conservation? The conservation implications of this are, how do we manage the population? Should we manage it as a collection of subpopulations, or should we return the population to a single continuous population? And how do you return a species to a single continuous population when they don't have a continuous habitat? Lions are currently in 28 different countries, so that to me sounds like a logistical nightmare. And the ways that you can try and manage that is through increasing connectivity or using translocation, or should we just stay hands-off and see what happens, and these different subpopulations could potentially inevitably become one of those 23 subspecies that we used to identify as the lion. So the suggestions that I have for management for Zambia, now that we know not only information about Zambia, but about this historical population as well, is that they should focus on maintaining genetic diversity by preventing the loss of genetic diversity within as well as between those subpopulations. Since there is natural movement happening between these two subpopulations in the west and the east, they should ensure continued natural dispersal by identifying those areas where the lions are moving and protect and protect those corridors by turning them into protected areas. And then looking even bigger picture, lions are a flagship species, meaning that as a large charismatic animal, research and conservation of the lion can impact a lot of other species within their habitat. And recent studies have shown that habitat fragmentation has a similar genetic effect across a wide variety of species, and not just mammals, but also birds and reptiles. So if fragmentation is affecting lions to this extent, it's likely having a similar effect of other species in their habitat. So knowing how habitat fragmentation has affected the lion population is not only important for lion conservation, but for wildlife across Africa also contending with anthropogenic changes to the African landscape. And with that, I would like to thank
the genetics program and the College of Veterinary Medicine. I'd like to thank all of my committee members. Thank you all for being here and helping me throughout all these years. And everyone in the DUR lab, especially David, thanks for being my office mate, putting up with me all these years. I'd like to thank Paula White from the ZAMBIA Lion Project, Leiden University, and UNT Health Science Center for providing information, protocols, samples, everything. Special thanks goes to Wes Brashear, Brian Davis, Ellie Armstrong at Stanford, and Laura Bertola at City University in New York. Without their help, I probably couldn't have finished this. They were very helpful. I'd like to thank all of the museums for allowing me to do destructive sampling of their precious collections. I know it's a big deal, and they really love all of their samples. I'd like to thank the African Lion Working Group for allowing me to be a member, and I may, because of being part of this group, I may actually have a say in the management of the lions in the next, see this research through to the end. And thank you to all my funding. So the one we got in trouble for is this guy right here, experiment.com. Thank you to all of you who actually contributed to that too. It was just a slap on the wrist. But Houston Safari Club, Dallas Safari Club, Safari Club International, Explorer Club, thank you for paying for all of my research. And then I would also like to thank this group of weirdos. Without you, I could not have made it through this. Any questions?